Hello, everybody, and welcome to the ALS United Mid-Atlantic Goals Podcast. I'm your host, Tony Heil, and I am here with ALS United Mid-Atlantic. We've been hosting this podcast for a little over a year now, and with over 50 episodes, we've had discussions with people from a wide variety of perspectives on ALS, from people living with ALS today, people who've lost someone to ALS, caregivers, advocates, researchers, and even people outside of the ALS-specific community who are working on other diseases, because united, we're helping to support and fund more science where we all win together. More funding for science research, for brain research, helps all of us because then we're investing in the cures of tomorrow for things like ALS, Parkinson's, Huntington's disease, etc. And we just showed that collaboration, that working together at our ALS United Mid-Atlantic Help and Hope Symposium that happened on April 9th here in Pennsylvania. We had a great panel discussion talking about how ALS, Parkinson's disease, AFTD, or frontotemporal dementia, and also Alzheimer's, all work together so we can learn things from each other. It was a really wonderful event. Some of the videos are on our channels right now that you can see. And one of our presenters is our guest today. His name is Dr. Eric Arnold, and I am very excited to talk with him today. He actually attended Haverford College, and then he got really involved in a PhD program at Thomas Jefferson University. He has talked about science like a long distance run. He understands that while we want cures tomorrow, it's going to take longer than that, and that's why it takes all of us working together. And he is working on genetic ALS at Washington University. So he knows a lot about collecting data, getting people involved in clinical trials, and making sure that you are empowered to help lead the next generation of ALS research. So, Dr. Arnold, thank you so much for talking today.
Yeah, thanks for having me. Yeah, I had a really great time at the meeting a few weeks ago, and so I'm excited to keep the discussion going.
As someone in ALS research and in science, how much of your time is spent going to conferences and meetings where you learn from others and network like that?
Yeah, that's a great question. I think every year I probably attend 4 or 5 conferences a year, I would say. And those are definitely really important. I think still in the age of Zoom, you get a lot out of meeting people face to face and seeing people and hearing about their research and then being able to see them immediately afterwards and, at a meal or during a break. So every meeting I go to, I feel like I leave with notes of people that I need to reach back out to. I got back in touch with you right after the ALS United meeting. New ideas, new people, and they do serve a variety of purposes. 1 or 2 meetings a year are more opportunities to meet people directly impacted by the disease. So the ALS United meeting also fell into that category. I think the discussion after my talk there was really interesting because it was very different than what I get at the researcher only conferences. And it's important to sort of really I feel like those really help to center me and ground me and, find purpose, renew the sense of purpose in my work. And then there's other conferences. So next week, I'll actually be at the Target ALS annual meeting in Boston. That is really research heavy. All of the, brightest, researchers in the field with the biggest, highest powered labs go and present their work. And so then I'm picking up new techniques and new collaborative ideas for my own research.
And I have found from my time here that a lot of people with ALS who have become involved in advocacy or research, they become really smart at understanding research. Our patients, I feel, they were never necessarily involved in science. They can be smart in other areas, but then they really put their efforts into understanding this. Do you find that you're meeting with people living with ALS, that they're very engaged in talking about the details of research?
Oh, yeah, absolutely. Absolutely. Yeah, to the point where maybe the first experience I had like that surprised me, but now I'm not surprised anymore. I expect to be getting really good questions, broader questions even sometimes, because people are reading broadly, and you can get a little more tunnel visioned in particular fields as a researcher sometimes, and we try not to, but I get questions about how my work might relate to some totally different finding that someone heard about. Maybe it's even in, like you mentioned in your introduction, something that came out as a new treatment for, Parkinson's disease, Huntington's disease, something that someone saw. Does my work relate to that? Things that wouldn't maybe come up in my own circle. So yeah, I find people to be really well educated, ask really good questions. Yeah, and I found that at the ALS United meeting too. We had a really nice discussion afterwards that covered a whole lot of a lot.
Really like that when we have these meetings because we have patients and caregivers who are listening and participating in conversations around research and science. Do you think that's a lesson for other diseases? Like, hey, maybe you should be listening to the lived experience more to help advance your science.
Yeah, 100%. I think yeah, I had a kind of a in grad school when I was at Thomas Jefferson University. My research focus was actually on a neuromuscular disease called spinal and bulbar muscular atrophy, and the genetics of the disorder are it's caused by a polyglutamine repeat tract expansion, which is the same type of mutation that causes Huntington's disease. So we went to meetings that included a lot of people with Huntington's disease. And I remember hearing someone with Huntington's disease start the meeting. This is the Hereditary Disease Foundation, I believe, and describe. I think someone asked, what symptoms affect you the most? And I remember the person really was focused on memory and cognition, memory being number one. And I remember to me, thinking about mouse models of Huntington's disease that we use, all the behavior tests are centered on the chorea, the motor dysfunction, and we look at other things too. But I remember that really stuck out to me that like hearing this person's experience, what is impacting them the most, what they care most about, maybe isn't the feature of the disease that that we focus most on when we're modeling it. So I've taken that with me, and I think the same thing applies every time I hear someone discuss what they're feeling. It helps to sort of reframe what you might think the most important things to focus on are.
I imagine for you, if you're using any kind of animal models, then you have to figure out what translates to a human. I talked with Dr. Zachary Simmons from Penn State Health Hershey Medical Center, and he was talking about how they use mice on treadmills, and he was
Yeah.
just pedaling along, and they measure their way of running.
Yeah.
But I imagine a memory test with mice. Maybe you could do something, obviously, with mazes, but you can't just ask them to do a question. Like if someone
Yeah.
has dementia, you might ask them, "Oh, who is the president right now?" A mouse probably is not going to answer that question, probably, unless
Probably
it's
not.
Ratatouille.
Yeah, exactly. Yeah, I mean, I think, you know, currently my lab, my new lab at WashU, we don't have a mouse colony. And I do think that there's been more and more scrutiny, I think to me, rightly on, if you're using mice, are they a good model for what you're trying to study? And not just taking it for granted that we do share a lot of DNA, and we're mammals, and so but taking a really hard look at the information you can get out of an animal. And so all of my work right now is on human stem cell derived neurons. Many of those come from samples donated from people with ALS, skin or blood samples that we can reprogram into stem cells and make neurons, and then trying to replicate and follow up on things in real samples, real human samples, blood, plasma, cerebrospinal fluid, or donated tissue. And I can imagine maybe incorporating mouse models at some point in the future of the lab, but I would want it to be for a very specific purpose. And there are sometimes you need a full organism with, we still can't model, the full complexity of all these organ systems working together, especially when you're talking about like drug delivery and things like that. You do need an animal model for that. But for some of the basic biochemistry, molecular biology, we've learned some hard lessons, I think, over the years as like a field that for age-related neurodegenerative disease, the animal models don't always recapitulate, or I should say what we learn in the animals don't lead us to effective therapies in the people with these diseases.
And that makes a lot of sense. And there's not a lot of other animals that you can use ethically or practically that would translate to humans. It's not like, oh, well, there's this other animal that's there that you could just easily use. It just doesn't exist.
Yeah, absolutely. And I think we are advancing like as a research community to be able to use the animal models in more specific ways that will answer more specific questions, like I mentioned about drug delivery being one, and assessing how, a drug that you expect something that you expect to be crossing the blood-brain barrier that you're delivering intravenously, that you want to get into the brain. Does it cause toxicity in another organ? Like questions like that. But for, I guess, ALS specifically, like a lot of my research is focused on the protein TDP-43, which many people have probably heard about. TDP-43 regulates thousands of human genes, and there's pretty poor conservation between what it regulates in mice versus human. So that's a really key part of ALS biology that we're studying, and the mice don't match what happens in a human neuron. So that's a real limitation right off the bat.
I need to make sure people understand your background a little bit because I did talk about your education. You're at Washington University. You are working on genetics and ALS. Because I want to talk about TDP-43, but just give me a quick rundown about what your expertise is to talk about genetics and ALS.
Yeah, and my background actually kind of splits into those two halves, and that's sort of where the lab is right now. So I already sort of gave a little background that in grad school as a PhD student, I was studying another neuromuscular disease, spinal bulbar muscular atrophy, and then as a postdoc, I began to study ALS, and my research was sort of in two categories. One being on the genetics of ALS, really the genetics of I trying to identify new genetic risk factors for sporadic ALS. So this was sort of a point that I was trying to highlight in my talk a few weeks ago, that even in sporadic ALS, genetics plays a big role. It's just in a more complicated way where you have interplay of multiple genetic risk factors all working together that contribute to ALS. However, if we can identify some of those risk factors, they still may be good therapeutic targets. And so as a postdoc working, and my work now, a lot of it is working with computer scientists, computational biologists, who mine the big data sets that we have containing all of the genetic information of thousands of people with ALS compared with thousands of people without ALS, newer tools, newer ideas, newer algorithms to tease apart differences in the genetics, new statistical associations between genetics of people with ALS versus without. And then the computer scientists need someone from the biology side to validate their findings, make sure that it actually that there's real functional biology there to study. So that was my work on that side, and that I'm continuing to do now. And then another big part of my work was studying, characterizing the functions of TDP-43. The reason we study TDP-43, as many people listening probably have heard, the challenge with sporadic ALS is that there is no, that every case might be slightly different, that the genetics and environmental epigenetic factors all contributing to the disease are different. And the fact that 97% of ALS post-mortem tissue shows this pathological hallmark of mislocalized aggregated TDP-43. That's really the only unifying hallmark we have that sort of brings all these cases together, suggesting that if we understand TDP-43 biology, we might learn something that is broadly relevant to sporadic ALS.
So, my understanding from talking with other researchers is then you can learn from people with genetic ALS. Like, they have a family history, and that way you can track what's going on with TDP-43, and that can impact anybody.
Yeah, I think that's fair. So the genetic ALS, the familial ALS, which these are often monogenic, highly penetrant cases. So it's a single, we know that there's this single point mutation in one gene that is sufficient to drive ALS. And that is the easiest thing to model in a laboratory setting because it's a single, you're not dealing with this unknown complexity of all of these genetic and maybe environmental risk factors. You can model in a mouse or in a neuron in a dish, comparing two sets, one that has a single mutation that that we know was driving a person's ALS, and then studying what happens downstream, which in those cases, except for in the SOD1 or FUS ALS, you see TDP-43 pathology. So we can use those cases to learn lessons that would apply to sporadic as well.
So I know that TDP-43 dysfunction is important. How do you know what TDP-43 is? Like, if I was a regular person and I walked into your lab or saw something, would I be able to identify what TDP-43 is as a new person to this?
What I would probably want to show you first are images, either in our neurons or in donated tissue sections where we can visualize TDP-43, most often with like antibody-based detection methods, where hopefully you would be able to see that what I've labeled either fluorescently, let's say, in a control sample would be localized within the nucleus of the cell. And then I should be able to find some examples. It won't be 100% of the cells. It might only be in 5% in the tissue section, where now that signal is not in the nucleus. It's completely outside of the nucleus. And in some cases, it will be forming these aggregates. You'll see little clumps. And that visual difference, I think, would be what would help you understand that, this TDP-43 protein is no longer in its normal location in the cell where it performs its function. And now it's in a different part of the cell. And not only that, but it's forming these clumps that are putting additional stress on the cell, would be the hypothesis. And I think that visualization helps to understand the two they work together, but the hypothesis that the reason that TDP-43 pathology contributes to or can drive this dysfunction that leads to motor neuron death is both through loss of its normal function, so it's no longer where it should be in the cell, but also gain of toxic function because you have these aggregates that are poisoning the cell essentially. And a lot of my work actually, I can be more specific by saying a lot of my work as a postdoc and now is actually on the loss of normal function side. What should TDP-43 normally be doing that it's not doing in ALS?
What have you learned about the loss of function? Is it normal for everybody, or is it that at a certain level it does this, or can you map that?
Yeah, so, and this is really right at the forefront of the field right now is that there, and this is something that I'm also trying to contribute to, but there's been a big push for biomarker development. Only in the last, the initial discovery published in 2016, more papers in the last 5 years describing the normal role of TDP-43 in regulating RNA processing. So if you think back to your earliest memories of a biology class, there's the central dogma is that our DNA encodes RNA and the RNA is translated into protein. And TDP-43 is acting at the level of RNA. So as your genome as your genes are being read, transcribed into RNA, TDP-43 is affecting the processing of that RNA. And when a given RNA is transcribed, this processing can ultimately lead that single RNA to encode multiple proteins called isoforms. And part of this is through a process called splicing. So TDP basically regulates which pieces of an RNA are going to be included or excluded from a transcript. And so TDP-43 is normally doing one of the key things that's been found is actually that TDP-43 is actually repressing the inclusion of certain parts of RNAs during the splicing process that turn that RNA dysfunctional. Most of the time, including that extra sequence that TDP-43 should normally be repressing, if TDP-43 is not in the nucleus, as it is in ALS, this new sequence will be inserted that kind of poisons the transcript, and it goes away, and then you don't see the protein that's being produced. Now, there's a small subset of the time where that extra piece that's being put into the RNA sequence will make it all the way into a protein that now has a new sequence that shouldn't be present in people without ALS or TDP-43 dysfunction. And people are looking into that very heavily to develop biomarkers. So that was a long way of saying that the field right now is really interested in biomarker development. And I think the earliest data would suggest that you do see evidence of TDP-43 dysfunction in most people with ALS. Probably not enough data yet to conclusively say when that happens prior to symptom onset, but there's definitely some suggestion that this happens before symptom onset.
It sounds like a game of telephone. Like, like there's a Simpsons episode where the teachers go on strike, and I forget the exact message, but it gets back to the main teacher, and she said, "We're going to get them for that purple monkey dishwasher comment." Like, it adds in something at the end,
Yeah.
and it's a whole other thing,
Yeah.
whereas normally it would just be the regular message.
Exactly. That's a good analogy, and our cells have really good quality control machinery that when these changes are being made, when these sequences are being inserted that shouldn't be there, it triggers the quality control systems that say this is no good, we're going to get rid of it. And that's why when these sequences are included, often it triggers just the cell to degrade that transcript, and then you lose the protein that it was supposed to encode. And it is only these rare cases where that new sequence makes it into a protein, and in some ways we can harness that for a good purpose to try to look for those peptides in biofluids, in cerebrospinal fluid or plasma. And I think that that's going to be, there's some promising targets already being developed, a lot of resources and thought being put into this. The dream of all of that work, I think, is twofold. Like, one, in a world where optimistically there are options, there are therapies that come out, these biomarkers will help us to track whether someone is maybe improving or not in a more specific way than the biomarkers we have now, which are good for measuring how many of your neurons are dying, but not necessarily specific to ALS biology. The other thing is that it would help people maybe to get diagnosed and put into clinical trials or on therapies earlier. So, like I said, there is some evidence that the current leading TDP-43 based biomarker that's been studied, for anyone who's curious, it's in HDGFL2. HDGFL2 is the protein. This came out of work from Phil Wong's lab at Johns Hopkins and Michael Ward's lab at the NIH, just moved to UC Berkeley. That, at least in Phil Wong's work, I believe they tested whether this biomarker was elevated in C9 ALS, in pre-symptomatic C9 ALS, and it was. That's, again, kind of one of those, like, fitting that theme of where the familial ALS can help, because that's really, with sporadic, there's no way to assess what's happening pre-symptomatically because we don't know until someone is sick. But with familial ALS, you can do things like that, like, try to learn these lessons of when does TDP-43 dysfunction occur, because we know that there's people out there carrying the mutation who don't yet have symptoms.
And since you mentioned RNA, obviously, this is maybe controversial, or it's been talked about for the last few years with mRNA technology with other diseases. Do you think that is a potential as a way of attacking or supporting ALS therapies or that kind of technology?
Sort of the way that ties most into ALS research, I would say, in terms of like oligonucleotide-based therapies, let's say, so that would include more broadly than talking about like the mRNA vaccines, is like these sort of oligonucleotide-based therapies would include tofersen, which is the first, effective treatment for any type of ALS, SOD1 ALS, and that is a sequence of chemically modified nucleic acids. So, I mean, conceptually it's the same as an mRNA. It's a sequence of letters that are complementary to a target of interest. And the way they work is by binding to RNAs that can either lead them to being degraded by the cell. So that's what tofersen does. It binds to the SOD1 RNA at the RNA stage, and it leads it to be degraded. So you're not making the mutant version of that protein. It also reduces the normal SOD1, but it's effective. To connect it with what I was saying before with the splicing, the function of TDP-43 in splicing, there are also antisense oligonucleotides that are being developed that essentially replace what TDP-43 is supposed to be doing. So TDP-43 binds RNA to prevent these splicing events, so the inclusion of this extra sequence that shouldn't be there. And you can actually make these oligonucleotide therapies that, instead of binding and triggering degradation of the RNA they target, you can bind, and essentially it's a acting like TDP-43 to just block this aberrant processing from happening. So that's really that's been, ASOs have been ASOs for all sorts of both familial ALS as well as some of these key TDP-43 target genes. Stathmin-2 and UNC13A are the big ones where there are clinical trials going on now to use this type of therapeutic strategy.
And it sounds, from my understanding, as not having your nearly 20 years of research and investment in this, like part of the problem with having that kind of mRNA technology or another kind of therapy like that for this is that you have to know when to be involved, like to send that information to your cells and what to say. Like if you're, I'm thinking about like exercising and like, oh, don't forget leg day. Like if you know that for this person, ALS is going to make their legs weaker first. If you get in too early, maybe they just have super strong legs and it just looked like the impact can be weird. You have to know exactly the right language to send and when to send it, or it might not be effective. Does that make sense?
Yeah, so I think that's one. Also, where these biomarker development is going to be so important, to have sort of like quantifiable measures that we can get from a person's blood or cerebrospinal fluid to monitor in a really clear way, like where they likely are in progression, how quickly things might be progressing, and even someday in the future getting even these molecular insights on, how functional is your TDP-43 at this stage, and then which treatment strategy will work best for you. But this sort of technology, the antisense oligonucleotides that first kind of worked as a miraculous treatment for SMA by the method where the ASO is blocking and changing the splicing of a gene. And then there was this great success, tofersen, with SOD1 ALS. And since then, there's been a few disappointments, most notably for Huntington's disease and for C9 ALS, which I think sort of, has made everyone, it's just sort of brought into focus sort of what you're saying, that it's not so easy. Like, people have a lot of theories for why the ASOs that didn't work ultimately failed, and some of it has to do with dosing and things like that, or the details about the target engagement and what you are targeting, I guess. So despite some early big breakthroughs with this technology, there have been some other disappointments that show that it's not so easy, and we really need like all of the information available to try to, to make these effective in certain cases.
Right, like if I'm taking regular medication for a headache, I know that I take that pill and when I have a headache, and that it will do whatever it does, and now my headache goes away. Maybe I sleep or drink water too, whatever it takes. But it reminds me now, as you're saying this, of being a father, and I have a 10-year-old, and if I tell him to do something one time, maybe it happens. But I have to tell him like 10 times, and then he will make sure to, clean up after dinner, or, he'll make sure to do his shower or whatever it takes. So you have to, if you're telling your cells to do something, and there's a lot of cells, you have to develop a treatment that is telling them consistently and at the right time and in the right way with the right language. So it's
Yeah.
not just we sent a message in a bottle, but we had to put all of this information in there and do it with this level of repetition to make it effective.
Yeah, and it's made even more difficult because the delivery method, sometimes is not so easy. It might require a spinal tap or something that has its own complications. So you can't always just say we're gonna give more frequent doses, because if it was a pill, then maybe no big deal. Other than, I mean, you can also get toxicity and off-target effects, but especially when the method of delivery, or would even there have been some clinical trials out of Clive Svensson's work at Cedar Sinai that, that requires surgery. Like, you're going to be putting something directly into the motor cortex. I mean, and so that needs to be like a one and done. But even when a clinical trial or a potential therapy is involving something like a spinal tap to be delivered intrathecally, yeah, it's tricky, yeah, because you have to really try to balance, and there's only so far you can get without actually doing the trial. I mean, that's, I guess, another like thing that I hear a lot, which is, I think, a nice way of putting things, that even the failed clinical trials teach us a ton, because like we were talking about way back at the beginning, the limitations of mice, you get so much information out of testing something in real people living with ALS, and everyone hopes that it will be effective, but when it isn't, that is a huge push forward for the researchers as well, just to know, you know, where to go next.
And TDP-43 was discovered just 20 years ago, is my understanding. So 2006, University of Pennsylvania. I don't want to
Yeah,
get into the whole
Virginia
science,
Lee. Yeah, yeah, yeah.
Virginia Lee, but I don't want to discredit someone else and get there on my end. But so figuring that out and then doing other science, it takes a long time. Is there potential that in 20 years from now, we know that TDP-43 can impact not just ALS but other things, that we could be doing a measurement at certain points in your life, and whether you're asymptomatic or any familial connection to a disease, and it's like, oh, this is probably a good thing to do at your 50-year checkup or your 60-year checkup, and see and
Yeah,
get moved into some sort of therapy.
I think for TDP-43, that's definitely a possibility. Yeah, not science fiction, because, um, yeah, and I guess I'll say, yeah, to avoid, yeah, I think in 2006, Virginia Lee's group, I think there was another group at the same time in Japan who, basically that discovery was that these aggregates that were present in ALS and frontotemporal dementia tissue, that the primary constituent of those aggregates was TDP-43. So, people knew before that there were these aggregates, but then that was the discovery that it was TDP-43. And the initial discovery of TDP-43, the reason it has the name it does, TAR, DNA binding protein, was actually the discovery that its discovery as a protein that binds HIV, actually. So it has this uh, whole other world of life, binding the viral sequence. Anyway, DP43 uh, pathology detected in 97% of ALS postmortem patient tissue, about 50% frontotemporal dementia patient tissue, also 40+% Alzheimer's disease cases, and there's a new, you can't quite call it a disease, but late neuropathological change, which is sort of like a late onset dementia, where that also has TDP-43 pathology. Also found in traumatic brain injury, CTE, like the NFL players are diagnosed with. So I think it is in the category of, if there are good biomarkers for TDP-43 dysfunction, I think it would come kind of right after looking at amyloid beta and tau, the plaques and tangles of Alzheimer's disease. It's something that would, yeah, not be a bad idea to check later in life if we have the tools.
Hey, I have to get my car inspected next month,
Yeah,
and I could see it being on your regular inspection for your health and your body, just to be checking whatever data you get on TDP-43 in your own cells, and then someone can say, "Oh, this is something we might want to direct you to as another kind of therapy before you have any symptoms of any disease."
yeah, yeah. I think it's definitely, like, yeah, you put a 20-year window on that. That's probably like the earliest I would go. And first, because we hope that, I think it's reasonable to think that within 5 years we will have biomarkers that are used routinely in clinical trials to measure TDP-43 function. And then as the technology progresses, yeah, and things become cheaper, more effective, maybe it would be added to, I think I could see it added to something like a panel of neurodegeneration associated changes that you might want to measure. I could see that. I like that vision.
Good, because my grandfather had ALS. We didn't have any specific other ALS diagnoses in my family other than him, as far as I know. But, personally, I would love if in 20 years that given my own history and my family, that I had these kind of scientific options available to me, and your science may end up benefiting me personally. So I want you to succeed.
Yeah, yeah, or yeah, and me too. I mean, that's the scary thing about sporadic neurodegenerative disease, and that's 90% of ALS and also Alzheimer's disease, Parkinson's disease, that um, it can impact anybody's family, let's say, at any time, or themselves at some point. So yeah, so everyone is sort of in this together. No one is immune. Um,
Right. Now, let me just bring one more thing up that your science and your descriptions remind me of, because when I first started here, I talked to an ALS researcher about this way of describing it, and it made sense to me. And it sounds like if you have too many tabs open on your browser, like too many programs open, and your computer's doing too many things, and then it crashes. Is that a one way of describing some of this issue with TDP-43 and your cells? Is like just things are not in the right place, and so like just like your computer can't function well because it's just got too much data going in the wrong direction.
yeah, it's an interesting analogy. I think I'm trying to connect it with TDP-43. I think with disease overall, I think it works like that the idea that your neurons, and this is where maybe the aging component comes in as well, because all of these, whatever, even if you have a familial ALS mutation, the onset of symptoms is happening later in life normally when compensatory mechanisms break down, sort of adds to the idea of this buildup of like energetic stress, like the demands on the central nervous system become kind of greater and greater, and then you start having crashes. I think the tabs analogy made me think about a hypothesis about ALS and applies to other neuromuscular diseases is the dying back hypothesis, that a motor neuron, a lower motor neuron, let's say, that's innervating a muscle, that the first aspect of dysfunction is disruption of that neuromuscular junction at the very end of the motor neuron, and then you get a dying back from there. And what happens earlier in disease is that the neighboring motor neurons will re-innervate that muscle fiber to try to maintain function, and that almost certainly is putting that motor neuron under a greater energetic load, a greater amount of stress. And so as motor neurons are trying to compensate for the ones that are lost, that's like each of them is adding more tabs to their browser as they're trying to, you know, keep the program running, and then eventually you get crashes from there. And that also fits with the way that symptoms typically spread in a manner that relates to the anatomy, the neuroanatomy, that symptoms are spreading in a way that suggests that the neuronal death is sort of propagating to neighboring regions, probably as the compensation is putting that area under more and more stress, and you're losing more neurons.
And I know that Dr. Simmons and others have talked about the impact of stress on ALS and on everything, and that's something that 20 years ago, I don't think we were talking about stress as a health issue so much. We have a focus on mental health here, which is separate from maybe this TDP-43 conversation, but maybe it's not separate. Maybe being able to talk about issues of stress, physical, mental, emotional, financial even, may end up having a positive impact on the progression of this disease then.
Yeah, I think for sure for sporadic ALS, which applies to most people, like nothing is off the table as being a factor that maybe changes the onset or progression a little bit. I've met people who are also pre-symptomatic with familial ALS in their families, and for those things, I still say, like, I'm in the Department of Genetics, and I believe that probably like your unique combination of genetic risk factors is what are probably contributing most to whether or not you get ALS, but not only. But I say that just because, especially for people on the other side have these mutations, you can also kind of drive yourself crazy wondering about how you try to modify your own life to prevent yourself from getting the disease, or to try to help yourself. And I think it's such a fine line, such a personal thing where you want to do what you can to minimize your risk factors and stress in some ways without going so overboard that you're not living your life when there probably are also things that you can't control, if that makes sense.
Yeah, it does. It's I see these videos on TikTok all the time, and maybe I need to get off TikTok. But they'll say something like, "We have like this napkin, and we're gonna drop a ball, and it falls through the napkin. And whatnot. But if we take four of them, now it's going to be fine." And I can see that your genetics predispose you to the napkins being weaker, but we know that it's the stress that leads to this, and we have stress that comes from our regular lives. You mentioned head injuries, or so. I'm not saying any of this directly
Yeah,
impacts
yeah,
ALS, but we know that age is a stress factor in life, and that
yeah,
obviously age is a factor when it comes to ALS in a lot of people. So if we know that you're predisposed to, that instead of four napkins holding up the weight, it's one napkin, so you're at a greater risk, but we can attack ALS from all these different angles by knowing more about all factors.
yeah, yeah, for sure. I think that, yeah, and I think the genetics and the environmental lifestyle contributions kind of go hand in hand, because as we figure out some of these more complicated genetic risk factors, that also helps with reanalyzing all the data we have to take people with a certain combination of genetic risk factors and then say what's different about their lifestyle versus someone else. For now, we're still in the point where we don't understand the complexity of how certain risk factors might come together to increase your chances of ALS. But as we learn more to be able to run these sorts of statistical experiments to say, like, people have started doing it. Actually, I think I mentioned this in my talk for C9 ALS, where the repeat expansion in the C9orf72 gene actually isn't 100% penetrant. So there are people who do not develop symptoms until very late in life, and then I think there's even some cases where they won't develop symptoms. It's highly penetrant, but not 100%. And I know that there's some work being done by geneticists um, to try to understand what is the genetic background of someone with the C9 repeat expansion who does or does not get ALS. And I think you can take the same, like, you could do the same thing on with lifestyle factors as well, because there's some lifestyle factors that have, I guess I would say, like, credible associations with ALS, like smoking and high intensity exercise for some reason, but no like big smoking gun yet, I would say.
Smoking tends to be bad for every disease. There
Yeah.
hasn't been a disease we found yet where they said, you know what, if you smoked more, you would probably. I have learned a lot from just talking to you now, and I probably could talk to you for the next 7 hours. But if you're interested in learning more about what Dr. Arnold has said in our Help and Hope Symposium, that video will be available shortly. And I think that we'll probably have more information from you. We'd love to share on our website and on our social media to talk about that. And so, Dr. Arnold, if people are interested in learning more about your work, do you have a website or place where people can learn more?
Yeah, for sure. So if you Google my name, or legally I'm Frederick Arnold, I go by Eric. I think if you Google me plus WashU, you'll find my page in the WashU Department of Genetics. I have a lab website, an early version of the lab website, but that should also have my email address. It should be pretty easy to find me, and I'm happy to hear from anyone for sure. I think also if you post my presentation, I think on my acknowledgement slide, I have my email address there. So yeah, hopefully I should be pretty easy to find.
And we'll put some of that information in the description here. If you're interested in supporting ALS research or ALS care, or you want to get involved in our organization, or if you know someone with ALS and you want them to get the resources they need for their house, like in-home care, accessibility, ramping, access to one of our multidisciplinary centers, access to social workers and nurses, and so many more services available to people with ALS here in Pennsylvania, New Jersey, and Delaware, please visit our website. Follow us on social media at ALS Mid-Atlantic. We have so many events, so many educational opportunities, and we're happy to connect you. Dr. Arnold, I have learned so much talking with you today. I feel like we can educate our families more, and I know that those living with ALS are going to be very inspired by your work. So thank you so much for what you're doing.
Oh, thanks. Yeah, it was a great discussion, and I'm definitely inspired by all of the people with ALS, affected by ALS, who are active in the community.
And together, We will end ALS with your help. Again, visit us, alsmidatlantic.org. Thank you so much, Dr. Arnold. I look forward to all of you being involved. Go back to our past episodes to learn more about ALS.
Thank you.